Hey everyone, it's Steven again today, and today I'll be going over Chapter 4 of the Cowan Microbiology textbook. Again, these are for preparation for my microbiology test on Tuesday, and these slides, just as a disclaimer, they were not created by me, they were created by the faculty at Sinclair Community College. So I'll just be going over them since this is what we were used to, this is what we were taught with. So I'll go over these to hopefully teach it back and maybe provide value to someone else out there. That being said, if you have any questions at the end or if you have any comments you want to put about things that I'm not saying correctly, I'd be happy to hear them, so just comment on the video below. Alright, so prokaryotic profiles. We're talking today about bacteria and archaea, and these are some of the more important things in our environment. So in my first video, I talked about the kingdom of Monera, which includes the bacteria and the archaea or prokaryotes, and so I'll be talking about those today. And there we go. All right, so this is just a picture of the end of a pin needle, I believe, and it just shows you how, even if you think that pin needle is clean, like it looks clean on the outside, it's a nice metallic surface and it looks nice and smooth. In reality, if you take a high-powered microscope, an electron microscope, and you look more closely, you can see that different microbes occupy that needle. So just kind of a cool thing to show you what's out there. So this is kind of what we'll be going over today. What are prokaryotes, the structure and function, the cellular envelope of the prokaryotes, the internal structures, and the different shapes and arrangements, and then we'll talk briefly about archaea because they're not as significant as bacteria are, but they are important as well. Okay, so the bottom line is that prokaryotic cells are the smallest, simplest, and most abundant cells on Earth. Compared to eukaryotes, they have no nucleus. So prokaryote comes from the Greek word for pre-nucleus or without a nucleus, and eukaryote comes from the Greek word for true nucleus. So there you go. That's the bottom line. Some of the other more significant differences: prokaryotes have a circular DNA structure without a nucleus, and they also lack membrane-bound organelles. As I mentioned in the first video, I talked about how eukaryotes are believed to have come from prokaryotes that phagocytized other prokaryotes to make them their organelles. And so prokaryotes, since they're more primitive structures or more primitive organisms, they don't have those membrane-bound organelles like eukaryotes do. But they do have cell walls, which is interesting. They have cell walls that include peptidoglycan, and this is how we test to see what kind of bacteria something is. We'll go into the different types of bacteria, but I won't go into it now, but that's one way in which we tell the types of bacteria is the type of cell wall that it has. They typically reproduce by binary fission, which means they basically bud from one another. Eukaryotes have multiple chromosomes as opposed to just one circular DNA. They do have a nucleus, membrane-bound organelles. If they have a cell wall, it's usually a simple cell wall. These are not, I guess I don't really know too much about eukaryotic cell walls, to be honest, but if they do have one, they're a simple cell wall. They reproduce by mitosis. Okay, so some of the external structures of a prokaryotic cell. These are the significant ones, and just excuse my shuffling paper here. Flagella, fimbriae, pili, and glycocalyx, and we'll go into these one at a time in more detail. Okay, so the flagella. This is an important aspect or an important appendage of a bacterial cell. Basically, it helps it move around. As you can see, the structure here. This particular bacteria looks like 3 flagella attached to it, maybe 2. I can't really tell exactly. But different bacteria have different numbers of flagella. Let's see, they are filamentous. They are used for motility or movement. They can either be monotrichous, amphitrichous, lophotrichous, or paratrichous. There's different arrangements of the flagella. So basically, if it's one flagella off the bacteria, one single flagella, it's usually referred to as monotrichous. Amphitrachus, that you can think of being ambidextrous. This is kind of how I think of it. I know ampha and amba are not the two same, what do you call them, prefixes, but basically an amphitrachus flagella means it is from both sides. So you can think of ambidextrous, amphitrachus. Lophotrachus means that there is more than one flagella off of the same side, so you might have three, like so. And paratrachus means that it might have multiple flagella surrounding the organism, excuse me. And here's some examples of that. So you have your monotrichus, one flagella. You have your amphitrachus, where it's coming from both sides like that. Lophotrichus, which is where you got a few from the same side, and then paratrichus, which is more, it's got multiple on multiple parts of the bacteria. And I guess it had all the labels, so there you go. So there are three main parts of the flagella. There is the filament, so that's the actual portion that sticks out of the bacteria and helps it move around. We got the hook, which sort of anchors, we got the hook here, which is where it kind of swivels around and helps the bacteria move. The basal body, which anchors the filament to the plasma membrane. So this is the anchor portion here, right here. And we have the fact that it rotates to spin, so it twirls around, as you can see by this arrow. And you can see the different portions of how it's anchored. You get the outer membrane and the cell wall. So actually, this is another important thing to mention here. I mentioned that there's different types of bacteria, which we'll go into in a little bit, but there's a gram-positive bacteria and gram-negative bacteria. And typically, a gram-negative bacteria will have an outer membrane outside of the cell wall, like so. Okay. Axial filaments. This is an example of a spirochete bacteria, as you can see from its little zigzag pattern here. Axial filaments, I believe, worked to tighten around the in the space between the cell wall and the cell membrane. So they tighten up and they sort of contract this guy, and then as they expand, that helps the bacteria move. But they're anchored at one end of the cell like a normal flagella would be. And there you go. Rotation causes the cell to move. Fimbriae are another appendage of the bacteria. They're stiff, bristle-like, and they help to attach bacteria to other things, including other bacteria. There's a few to hundreds on one cell. They enhance colonization and increase virulence. So as I said before, they help them attach to each other, but they also help them to attach to other cells that they're trying to infect. So that's what we mean by when we say virulence, is basically the ability of it to infect other cells and enhance its survival, essentially. Pili is another appendage that's on the bacterial cell. It's longer than Fimbriae. There's only 1 to 2 per cell as opposed to many of them. They're made up of these hollow tubes for protein called pilin, and they are used for the partial transfer of DNA from one cell to another. So I believe, say like a bacteria is resistant to an antibiotic, say penicillin, and one guy wants to transmit his DNA to another bacteria so that he's also resistant to penicillin. This is one way they can do it, is through these pili, and this is how they communicate. So you could think of these as the little cans with the strings attached to them that you'd communicate with your neighbor back in the old days. Let's see. Another external structure is called the glycocalyx. It's a covering, it's like a sticky covering surrounding the cell wall, very viscous and gelatinous. Let's see if there's anything else to say. It's made of polysaccharide or polypeptides. Remember from my last video, polysaccharide means many sugars or many proteins, or many amino acids, of a protein. These are kind of what they look like here. You have this sticky substance around the Streptococcus pneumoniae. That's the bacteria that causes pneumonia. And you get the slime layer on here in this guy. Different types of glycocalyx. We have a capsule glycocalyx. a very thick layer firmly attached to the glycocalyx, and these are two types, the capsule and the slime layer. Slime layer is thinner, loosely attached to the cell wall. And I believe we'll talk about biofilms in just a second here. They prevent things like phagocytosis and dehydration of the bacteria cell. So this is how they help themselves sort of evade our immune system, is by secreting these glycocalyxes. It prevents phagocytosis, so it helps prevent things like neutrophils and macrophages from digesting them, prevents dehydration, maintains nutrients, and facilitates attachment. So biofilms, this is a part of the book where it talks about biofilms. Basically, it mentions things like, say you're inserting, basically whenever you're inserting something into the body, you have to worry about things like infection. And one of those instances is like a catheter. And what will happen is those bacteria will form biofilms around that catheter, and this has been a problem with as far as keeping patients away from infection, is those biofilms, because it's harder for those antibiotics to penetrate that. And someone can correct me if I'm wrong here. I'm not exactly sure on these. I'm not an expert. I'm just going from what I read on. Here's an example of a dental plaque. So this is another way that biofilm, or that glycocalyx layer, sort of manifests itself as the plaque on our teeth. That's why if you don't brush your teeth for a day, or sometimes less than a day, then you'll feel that plaque on there, and that's what that's coming from. It's the bacteria. The cell envelope, so has 2 or 3 basic layers. So the cell envelope is sort of the overall term for the covering surrounding the bacteria cell. So for eukaryotes, we have our cell membranes, which are composed of the phospholipids. Bacteria have it as well, but they also have more layers. So they have a cell membrane, but they also have a cell wall, which is very characteristic of bacteria. And then if they're a gram-negative bacteria, which I tried to mention earlier, they also have an outer membrane that's outside of that cell wall. And those are important it's an important thing to know, because if we're infected, we need to know what kind of bacteria we're trying to treat. So this is sort of that breakdown of that cell envelope. Here you have your inner cell membrane, you have your peptidoglycan layer. Peptidoglycan is part of the cell wall. That's the main component of it. And an outer membrane. So, gram-positive and gram-negative. The difference is that in gram-positive bacteria, we have this cell wall that's very thick like this, thick of peptidoglycan, and then we just have the cellular membrane under it. Versus for gram-negative cells, we have an outer membrane, and then we have that peptidoglycan layer in the middle of the outer membrane and the cell membrane. And so this, the way we tell, or the way we distinguish between a gram-positive and a gram-negative bacteria, is by this process of staining. So if we stain the bacteria and it turns like a purplish color, that's usually characteristic of a gram-positive bacteria, because that dye is being absorbed in the peptidoglycan layer. Now with a gram-negative bacteria, that dye doesn't really penetrate as well. So what will happen is you'll have a pink layer. And that's sort of more relevant to the lab, but just so for your awareness. Here's another view of that cell envelope. Another important aspect of the cell envelope is these teichoic acids. These are, if I remember correctly from immunology, these are markers for things like the macrophages and neutrophils to attach to, that sort of mark the bacteria as a foreign organism that the body needs to get rid of. So if they see these things, this can be a marker for destruction within our own bodies. Here you have the gram-positive bacteria. So this very thick peptidoglycan layer with these teichoic acids sticking out of them, and the cell membrane just sitting underneath. Versus the gram-negative bacteria, you'll have this cell membrane, a smaller peptidoglycan layer, and then an outer membrane. One of the things that our teacher mentioned was that these gram-negative bacteria tend to be more evolved because they are able to evade our immune system a little bit better. The cell wall composition. So That peptidoglycan layer is made up of repeating subunits called glycans. These are disaccharides, as mentioned from or as brought up from our previous video here. Just gonna make sure I'm not missing anything. Okay. Made up of these different types of disaccharides, this N-acetylglucosamine, or NAG, and N-acetylmuramic acid, NAM. And then, let's see. These polypeptide subunits are a tetrapeptide side chain and peptide cross bridge. Just make sure I'm not missing anything here. This is basically just telling you how the proteins, so it's made up of these proteins and disaccharides, right? So we have this is how these peptides form. So the function of the cell wall is to prevent rupturing. It's that main outer layer, especially in the gram-positive bacteria, helps maintain the cell shape, and is an anchor for flagella. And as you can see from that previous slide, we have the NAG and the NAM sort of connected to each other. And then these different layers attached via these tetrapeptides, so the amino acid cross bridge. So basically the role of the proteins are to connect these sugars together in the different layers, whereas the sugars are more in line like this. Is anything different to say here, really? Cell wall, cell exterior. Just another view of the gram-positive and gram-negative bacteria. Peptidoglycan. Again, this is how we distinguish between the two major types of bacteria. The cell wall makes some bacteria susceptible to antibiotics, so this is the main thing we attack when we attack bacteria, is the cell wall. It's an easy target, because if that ruptures, then the whole cell dies. All right, and then more on the gram-positive cell wall. So it consists of 60 to 90% of those peptidoglycan. It consists of a 60-90% peptidoglycan layer. It's got these techoic acids, which I mentioned before, for antigen specificity, but they also provide stability for the cell wall and an acidic charge. Let's see. Yep, and those techoic acids there again is another view of that chart. Versus a gram-negative cellular envelope consists of only 10 to 20% peptidoglycan layer. It's got a periplasmic space. Let me just look through my notes real quick. Active in cell metabolism and a reservoir of digestive enzymes and transport proteins. So this is I don't want to speak out of term here. The periplasmic space here, as you can see, is between the cell membrane and the peptidoglycan layer. Right there. Okay. Active in cell metabolism and a reservoir of digestive enzymes and transport proteins. And then you get your outer membrane, which is characteristic of those gram-negative cell envelopes. The outer membrane itself is made up of a lipid bilayer. It's got those lipopolysaccharides on the outside of the outer membrane. The lipid A portion is called endotoxin. This is some of the symptoms of that endotoxin. The porins are the protein channels that regulate the movement of substances into and out of the cell, including antibiotics. So this is another sort of defensive function that gram-negative bacteria have over gram-positive bacteria, is these porins that sort of give it another defense mechanism. Another view of that chart there. And going to the plasma membrane, which is characteristic of both the eukaryotes and the prokaryotes. So it's the barrier between the cytoplasm and the environment. Multifunctional. It's made up of a phospholipid bilayer, just like ours, and then proteins. And the way that this is sort of modeled is it's called the fluid mosaic model, which just means that it's basically a layer of many different parts, and it's sort of in fluid motion. It's never really stiff. There's more to read about that. I don't know a lot about the basis for it, but It's just what it's modeled after. So the functions of the plasma membrane are to regulate the movements of materials into and out of the cell through those proteins called porins, help synthesize cell wall components, plays a role in DNA replication, secretes proteins, and carries on respiration, captures energy as ATP. Okay. Again, this is the phospholipid structure, which we mentioned before. So we have a group here with a phosphate head, the hydrophilic head, and then we have our hydrophobic tails. Whoops. The phospholipid bilayer, lipid tail, proteins, peripheral proteins, which are part of the membrane. Some of those proteins are used for enzymes and metabolism and support. And then you have the integral proteins, which are more sort of transmembrane. They go from inside to outside, like the porins. And here's sort of more on the fluid mosaic model. So you have these different, you have the phospholipids themselves. You have the glycolipids, which stick out of the membrane here. Same thing with peripheral proteins. This is the outside of the cell. You have these integral proteins like the pores and the channels. And there you go. It's another view of it. So one way that these cell walls are damaged, so this is how we sort of fight infection, is the lysozyme digest disaccharide and peptidoglycan. Let's see. I'll be honest, I'm not sure what the lysozyme role is here. Probably should have paid more attention in class here to this portion. Penicillin inhibits peptide bridges in peptidoglycan. So those bridges are important for maintaining the structure of that peptidoglycan cell wall layer. That being said, there are some bacteria which can break down penicillin, and those are the ones that we're concerned about, those antibiotic bacteria. You have the NAG and the NAM, which are the sugars here, and then the amino acids that connect them in the stacks like that. That's another view of it. Alcohols can damage the membrane, like detergents and these polymyxin antibiotics. They cause leakage of cell contents. Again, these are the main components that we're trying to attack when we're attacking bacteria in our bodies. Some non-typical cell envelopes are mycoplasmas. They naturally lack a cell wall. Membrane is stabilized by sterols, and they're pleomorphic. And I believe one of the examples of these is tuberculosis. This is a bacteria that tends to hide in our macrophages, and it's very difficult for us to get rid of. It tends to manifest itself and then hide again, and manifest itself and hide again, and that's why it's so difficult for us to get rid of. Acid-fast bacteria. So these are mycolic acids, waxy lipids, and very slow growing. This is another thing we can test for in the lab, is these acid-fast bacterias, because they secrete a certain these waxy lipids, I think. I think I probably didn't take enough notes here as well. The cytoplasm, same thing as in our cells, 80% water. Enzymes, lipids, carbohydrates, and inorganic ions. It's got these structures of these nucleoids. Let me just look real quick. Ribosomes and inclusions. Ribosomes being the main function of our protein. There are protein factories. Okay, and then the nucleoid. In contrast to our eukaryotic cells, they are not surrounded by membrane, so they don't have a membrane-bound nucleus. Actually, they don't really have a nucleus at all, right? So Bacterial chromosome are single, large, and circular, made up of a double-stranded DNA. And we also have different strands of proteins called, or different strands of DNA called plasmids. These are smaller circular DNAs, and these are extra chromosomal. And these, I believe, are ways that, so if you're trying to get a bacteria to produce something for you, what you'll do is you'll put these plasmid DNAs into the bacteria. And what will happen is those plasmids will get absorbed into the bacteria's main DNA, and those will dictate which proteins the bacteria produces. The ribosomes, again, are the site of protein synthesis. These are sort of the components made of ribosomal RNA and proteins. And they are two subunits which differ from eukaryotes. So a smaller subunit and a larger subunit. Typically what will happen is when you have a DNA, it'll encode a messenger RNA. And then in order for that messenger RNA to create proteins, it'll sort of go through this ribosome like so. And that is how it'll translate into a protein. Like so, you have the messenger RNA, and what will happen is these transfer RNA will bring different amino acids based on what the messenger RNA codes for, and you'll see this polypeptide chain starting to form here and make our functional protein. This is sort of a breakdown of where the different antibiotics will, this is how they'll inhibit bacterial function. So you'll have this chloramphenicol binds to the 50S portion and inhibits the formation of the peptide bond, so you won't have a growing peptide chain right there. The erythromycin will bind to the 50S portion, prevents the translocation movement of ribosome, so prevent the ribosome from moving along the mRNA. These tetracyclines will interfere with the attachment of tRNA to mRNA ribosome complex. So without the tRNA bringing the amino acids here, the protein can't form. And these streptomycins will change the shape of the 30S portion right here and cause the code on mRNA to be read incorrectly. So these are some of the functions or some of the ways that antibiotics will prevent bacterial growth inside of us. Just get a water break 1 second. Inclusions are within the cytoplasm. They are storage components for nutrients like phosphates and lipids and polysaccharides and sulfur. I'm not sure what the analogous portion in our bodies would be. The actin cytoskeleton, so these are long chains of protein arranged in helical ribbons under the cell membrane, and they contribute to the cell shape found in rod-shaped and spiral bacteria. And so these are basically the things that help give it its structure, give the bacteria its morphology. Endospores are basically they're formed in the cytoplasm of some gram-positive cells like Clostridium and Bacillus, such as anthrax. And what these endospores are is that they're the bacteria in like a resting state. So it's more of a state that the bacteria wants to be in if it just wants to survive. It's not really in its living state. It's dehydrated. It's very resistant to heat, cold, desiccation, chemicals, and radiation. It's not a form of reproduction. It's just a form of survival, essentially. One bacteria cell converts to one endospore. Versus when it's in the vegetative state, it's its normal active form. And sporulation is the process of forming endospore from a vegetative cell. I think it'll show a picture of that in a little bit. Germination is where the spore cell returns to the vegetative cell. So, again, sporulation is the process of forming an endospore from a vegetative cell. So sporulation is like survival, right? Germination is where the bacteria wants to cause germs, and so it returns to a vegetative state. Sporulation is survival. Germination is infection. And this is sort of a breakdown of how that endospore is formed. So we have a bacteria where it's talks about the spore septum begins to isolate the newly replicated DNA. So this newly replicated DNA is starting to separate from the main part of the cell. Talks about the plasma membrane starts to surround the DNA and isolates that DNA by itself. The spore septum surrounds isolated portion, forming a forespore. So you have two membranes now around this DNA that's about to get out of Dodge. The peptidoglycan layer forms between the membranes. So you have this guy. The spore coat forms, and the endospore is freed from the cell. So you can think of this as sort of like an escape pod, essentially. So like this ship's going down, like this guy just wants to get out of here and survive. So he gets into his escape pod and he gets out of Dodge. And this is sort of a picture of that happening in real life. Archaea, the other prokaryotes, which they're not mentioned as much, but they are single-celled also, and they're simple organisms that are prokaryotic in general structure, proposed to be in their own domain, and they are the most primitive of life forms which appear to have descended from the first cells that originate on the Earth. They're called extremophiles because they are adapted to live in environments that would be deadly to others. So high pH environments, high temperature environments, or on the opposite, the low temperature environments. They're basically the last things that would be living on the Earth, probably. Excuse me, those are some examples of the different types of archaea, the methanogens, the halophiles, psychrophiles, cold conditions, hyperthermophiles, high temperatures, acidophiles, low pHs, and sulfur reducers. There's more on the shapes and arrangements. So the morphology of prokaryotes, we have different structures of them. We can have circular structures, which are called coccus or cocci, bacillus, which are more like rod-shaped, spiral. So you have they're literally a spiral, and the size range is 0.2 to 2.0 micrometers in diameter. Here's some examples. You have the cocci, the bacilli, the rods, and you have the spirochetes, or the spiral shape. Let's go back. Yeah. These cocci can be arranged in different formations. If it's a single can be a single cocci, can be a diplococci, which is two, or if it's a chain like this, can be streptococci. I have to pull up my notes to get the full. If it's a cluster like this, it's going to be a staphylococci. Let's see if it'll bring that. Okay, there we go. Diplococci, streptococci, and is in a chain like that. A tetrad, so you got 4 together. A sarcine, which is 8 together, like 2 tetrads. And staphylococci. So again, diplococci is 2, streptococci is a chain, a tetrad is 4, a sarcine is 8, and a staphylococci is like a bunch of berries all together in one. You can have these different arrangements of bacilli as well. So here up top, we can have a single bacillus. We can have a diplobacilli, a streptobacilli, which is a chain again, so strepto is going to be in chain. Or we can have these coccobacilli, which are sort of semi-cocci, semi-bacilli that are together like that. And the types of spiral bacteria. You can have a vibrio, which is sort of one little bean-shaped bacteria like that, like so. You can have the spirillum, which is more just one little swirl. Or you can have the spirochetes, which are more sort of zigzag all around. That's the end of the show. Again, if you have any questions or if you have anything that I may have missed that you think might be beneficial to know, please put that in the comments, and I'll see you next time. Thanks.